percent desktop. Yeah. Can you see now?
Yes, sir.
Yes, sir.
Okay, that's good. Yeah.
Sir, you can put into the full screen mode.
Yeah, I will put it in full screen mode. No problem.
Yeah.
It is now. It is full screen mode.
Yes, sir. So welcome back to the second session of the first day. And in this session, we are having the two presentations. First talk from the Dr. Shyam Singh Phartial, and second is the Professor R.P. Singh. So it is my pleasure to introduce the first speaker, Dr. Shyam Singh Phartial. Dr. Shyam Singh Phartial holds the PhD degree in the forestry from Forest Research Institute, Dehradun. He received postdoctorate fellowship in 2001 from Vision University, Bristol, Brazilium, to work on the soil seed bank and ecology of the Borsal city forest. After returning to the India, he obtained the CSIR award to the Independent Research Associateship to the work in the seed biology of the medicinal plant and forest research institute, Dehradun. He is the recipient of the prestigious JSPS postdoctorate fellowship from the Japanese Society for the Promotion of the Science and the Work at University of Japan. He received the world-renowned Humboldt Fellowship. Currently, he is working as an associate professor in the Nalanda University. Before joining to the Nalanda University, he is working as assistant professor in HNB Garhwal University. Dr. Phartial is published so many research papers in the reputed national and international journal. Today, he will deliver the talk on the seed dormancy, germinations, and the climate change. I believe no one can explain this topic better than him. So now I welcome to the Dr. Shyam Singh Phartial to this virtual platform, and welcome to you, sir.
Thank you, Abhishek. Thank you for nice introductions. And as Abhishek said, seed ecology, like India doing very good in agriculture science, because agriculture science we lot know about the seed breeding and having a new variety of the seeds and every aspect of the seeds. But what happened to the wild seeds? We have very poor knowledge about the wild seed, except few ornamental plant which can be used for the floriculture, some horticultural plants, and some plants which are wild relatives of the agriculture crop. But we have more than 360,000 plant species, seed plant species in the world. Do we know how they germinate, what kind of, when they disperse, when they mature, when they disperse, how they disperse, how long they go away from the mother plant, when they disperse, when they germinate, how many days they take to germinate? Let's have this first figure. Anyone, can you tell how long from, can you see my mouse, my arrow?
No, sir.
Yeah. So from this first picture to the last picture.
Yeah. Now we can see, sir.
Yeah. How many days it can the seed can germinate? You use the moong dal at home. You sprout every day. You sprout the moong dal or the chana dal. How many days it took from this dry seed, then imbibe seed up to these portions or up to these portions. Any guess? It took almost 1 year, more than 1 year. So when you work in a wild seeds, it's not a working like agriculture seeds. You sow in the morning and by evening they get germinate. So that's why how the climate is going to impact them, that is more important to know about that. So look at this scene. This is a extreme weather conditions. You can see, okay, there is a extreme dry spell and it's going to kill everyone. Then this is a flooded one. We have a flood everywhere. Water, the extreme one had a flood and other had a dry spell, no moisture. And this adult man or farmer, which is now have already worked hard, can develop the immune system to sustain or to survive in both dry as well as in flooded weather conditions. This is my daughter. A few years back we took. Can you see this? A baby can survive in these conditions if you
Sir,
leave the baby. Yeah.
Sorry for interrupting.
Yeah.
The slides are not moving, sir. We are seeing only the first slide still.
Okay. I don't do.
Now we
Yeah.
can see, sir. Now we can see.
See?
Yes,
Yes,
sir.
yes.
Now we can see. Yeah.
Yeah.
Thank you.
So this old man can, because adult life and this baby may or may not survive these extreme weather conditions. Now take a parallel of these old age and then just a child age or a newborn baby or something. This adult plant may or may not survive, or most of the time survive. Either it is having a dry spell, the root system will dig the moisture from the deep soil surface and survive. And when they're flooded, the majority of these plants are surviving. What about this? Neither the flooding nor the dry spell, this plant, this baby plant will survive. So there is something background noise. So what I'm trying to say from this picture, whenever the climate change research happen, the climate change research happen only on the established plant, how their growth pattern, their yield and everything. But we forget which is the seed can be one of the basic tool to which can be, that manipulated by a microclimate level changes, a little higher content of the soil moisture, or little the lower side of the moisture content, or little higher temperature or little lower temperature can hamper the germinations and dormancy pattern of the plant. But what happened, majority of climate change research either happening through the remote sensing, policy label, adult plant label, adult life, but hardly we look how these baby plants or a human being as a baby. That's why I took a parallel of these two things, how these baby plants will impact. So, and my talk will be more or less roam around this topic, the global change biology. The three of the co-author of this paper, Jeffrey Wallach, Sethi Hyati, and Peter Poslord, they are my co-author in other paper. And they written this paper, and this paper is already cited more than 600 times. So climate change and plant regenerations from seeds. So at the core of the plant regenerations, the temperature and water supply are a critical drivers for seed dormancy, either to initiate the seed dormancy or to break the seed dormancy or to start a germinations. Because a dry seed cannot germinate. But once they get appropriate temperature, appropriate water supply, they come out from the dormancy, or the dormancy can be introduced, induce on them and then germinate. So global climate change is altering these environmental cues and will preclude or delay or enhance the regenerations from the seeds. Means, some seeds are going to disperse during the monsoon and they germinate during the monsoon, because during the monsoon the temperature is optimum, the moisture is optimum. If the monsoon will be little bit fluctuate from early or late, then this whole process of the seed germinations and dormancy break will be hampered. So look this box. The factor in box being most affected by the climate change, the seed dormancy. Seed dormancy, the dormant seed has a temperature and moisture. It is in dormant. When there is no optimum temperature, no optimum moisture, it will remain in dormant. As soon as it get a optimum light, optimum temperature, day length, and all kind of the moisture, if it is in a fire or a smoke area, like in Australian Mediterranean region, the smoke will induce the dormancy, and then the anox, and then it becomes a seedling. So these two phases are going to be most hampered or affected by the climate change or extreme weather. Now, Dutch species has a characteristic germination season. Yeah. The time of the year of seed germination is short for many species. Some only germinate in autumn. Those species which going to be like winter annuals or summer annuals, they have a different, like the wheat cannot germinate in a spring. Wheat need to be sown in during the winter, and then rice need to be sown in a late spring or summer. Or summer. So some germinate in autumn, some germinate in spring, some germinate in wet season, like in a monsoonal pattern, depending on the where you are, either you are the northern hemisphere or in the southern hemisphere, or the seeds or plants are in a tropical region or a temperate region or subtropical region. In contrast, germination season for others is long throughout the growing season. Majority of invasive species, whenever they get a little bit moisture content, or they germinate at a wide range of the temperature and wide range of the moisture content. And even some throughout the year, you want to sow your sprout, your moong dal anytime, they germinate any point of the time. So how this time control in nature? The important factor to control the timing of a germinations in nature is a seed dormancy. As I said, this is a Cardiochitinum giganteum, a species from the Himalaya. It dispersed its seed in October 2006 and it germinated. It took seedlings come out in April 2008, almost like 18 months. How? Majority of time we use only one temperature when we do the seed germination ecology or seed germination studies. We forget about. That's why the seed ecology is very fundamental tool for the study of a climate change. Like glaciologists, they are measuring the depth and then volume of the ice. The other, they are measuring the different the chemical component of the in atmosphere. Similarly, at the plant ecologists, we have, this is a basic tool to study the climate change, how they are going to impact to the plant regenerations. So what is seed dormancy? Seed dormancy is when seeds are incubated under the favorable conditions for germinations. If they are non-dormant, they need to germinate, but they do not germinate. Means you given everything. Means your parents give you good, admitted you in a good school. They given a good book. They given you every possible the tool to study, but you are not studying. Similarly, we given appropriate temperature, appropriate moisture, appropriate light, but still seed is not going to germinate. That state is called a dormant stage. Generally, we take 4 weeks as a thumb rule If any seed is going to germinate within a 4 week, we say it is a non-dormant. If it take more than 4 week, we say it is dormant seeds. Dormant seed usually take much longer than 4 weeks, as I said. And seed dormancy simply means seed are not germinating. However, many things could be occurring in the embryo or including growth. There is some hormonal disbalances over there. There is something kind of morphological or morphophysiological things going to happen inside the seeds. So the lack of germinations is a seed problem. It is not an environmental problem. Sometimes it's okay. This environment is not meeting. We're not given a proper moisture or proper temperature. No, if seeds are dormant, if seeds are non-dormant, then this temperature, moisture can play a role. But if seeds are dormant, it is not an environmental problem, it is a seeds problem. Something going inside the seed which is not permitting it to grow. So if any one of you are interested in seed, the Carol and Jerry Baskin, I'm fortunate to work with them. A classification system of seed dormancy. They published this paper in 2004. It is one of the highly cited paper. I think more than 2,000 times it is already cited. And then they published this book, Type of Seed Dormancy and Kind of Seed Dormancy. You can go, and this is an encyclopedia for as a seed ecologist or for the regeneration biologist. We call it our Bible. And they are like God for seed ecologists. And recently they published this paper. Unfortunately, I was one of the reviewer of this paper. They discuss from the orchid seeds to the the palm seeds, and from parasitic plant seeds to the all kind of. And this is another great those who are interested in molecular aspect of the seed dormancy and biochemical aspect of the seed dormancy. The seed biology places a very good resource to dig down. So now, based on, because I will come again with the climate, I will go climate and these together. So before to understand the germination, dormancy, and climate change, we need to understand how we're going to classify, because majority of, because you are an environmental science, majority of students may be in biological science, maybe you heard about the seed dormancy during your intermediate or maybe some botany classes of the semester 1 or 2, or very superficial knowledge about the seed dormancy. I'm going to a little bit deeper. So as per the Baskin 2004, 2015, and 2021 classifications, they've done a hierarchical classification system of the seed dormancy. They divided the seed dormant in a 5 broad category: morphological dormancy, physiological dormancy, morphophysiological dormancy, physical dormancy, and combinational dormancy. Means any seed, if has an underdeveloped embryo, but the underdeveloped embryo is need to be developed before the seed to germinate, we call it a morphological dormancy. If the seed has a low growth potential of an embryo which cannot overcome mechanical constraints on a covering layer, maybe physiologically that seeds may not that much ready to penetrate the seed coat and come out from the seed coat, that we call it a physiological. It can be due to the hormone, it can be due to the any kind of the gene signaling or any biochemical point. Because I'm an ecologist, I'm not going too deep on a bio. Then morphological dormancy is a part, it is combinations of the both. You have a morphological dormancy, you have a physiological dormancy. Physical dormancy means a seed is water impermeable, of have a palisade or palisade-like cells in seed or fruit coat, means a seed coat is not permitting water to enter inside the seed, because until and unless the water will not reach to the embryo, the seed cannot germinate. The germination process will not start. Then combinational dormancy means a seed has a physical plus physiological dormancy. So I will explain them in detail. But I will give you one. Majority of you may learn from the old school of books, like exogenous dormancy, endogenous dormancy, mechanical dormancy, chemical dormancy, thermodormancy, photodormancy. No, this new classification system does not recognize mechanical dormancy, thermodormancy, photodormancy. They say, okay, either seed is not germinating due to the light, due to any chemical component, or due to some mechanical resistance. If it is going to imbibe the water, that means it is something happen with its physiological problem. So they keep all the mechanical dormancy, chemical dormancy as a part of the physiological dormancy. So whenever you read in old style book, especially the Nikolaev's or Harper's classification system, please think that is they are talking about the physiological dormancy. So now how we categorize, I will, because majority of students are over here, I thought it is better to make it for students to get some interest, and they can take a baton from people like me who can become a next generations of the seed ecologists in India. How we categorize the seed dormancy? So if any seed has a fully developed embryo and 50% seed germinate within a 30 days at appropriate temperature or moisture conditions, we call it as a non-dormant. If you are, some of you are working in agriculture seeds, sometimes if agriculture seed take, instead of 3 days, it take us 7 days, to agriculture people say, oh, dormant seeds. No, as per the seed ecology, overall seed ecology of the wild plant, we say no. Anything which is going to germinate within a 30 days, it is non-dormant. But for the agriculture, they need to grow or yield to reduce the timing of the yield shorter. So for them, that is a delay in germination, not a dormancy. If the species or seed has a fully developed embryo, I will show the diagrammatic scheme how we call the underdeveloped and fully developed. So if it has a fully developed embryo and 50% germinations happen within a 30 days, not happen, if the seed take a more than 30 days to germinate, we call it is a physiological dormancy. If has an underdeveloped embryo and it germinate within a 30 days, we call it a morphological dormancy. If underdeveloped embryo but take a more than 30 days, we call it a morphological dormancy. So this is a combination. Embryo is dormant, underdeveloped, but also has a physiological dormancy. So we call it a morphophysiological dormancy. But when the seeds not at all taking the water, maybe you sometimes when you take this sprout and other things, all of a sudden 1 or 2 grains are still very hard. So they are physical dormancy because the seed coat is not permitting the water to inside the seeds and they are not imbibing the water. So again, going to one by one, the morphological dormancy. So this is the Lonicera cerula, honeysuckle. Majority of, if you have the northern hemisphere, Lonicera cerula or the honeysuckle is like a berry, blueberry. We also call it a blueberry. So this is one of a very famous horticultural fruit in Canada, US, Europe, Japan. And we published this paper on Lonicera with the Carol Baskin and Jerry Baskin during my JSPS fellowship during 2009. So what happened? Underdeveloped embryo means the embryo is underdeveloped, but before the radical come out, that embryo need to develop inside. So how it develop? It need optimum temperature, moist substrate, light or darkness depending on the physiology of the, and this happen within a 30 days. If you are not looking this embryo phenology, you may categorize this species as a non-dormant species, because the definition of the non-dormant species, any seed is going to germinate within a 30 days, that is treated as a non-dormant. But in this case, the embryo is dormant, not a seed. So the embryo is developing inside the seed. So we call it a morphological dormancy. Look, this same species, I cut the on soon after the collecting the seeds. It took a 0 day, on 0 week. Then within a 1 week, the embryo developed. Then within a 2 week, embryo fully developed. And by the 3 week, embryo fully developed and radicle come out. So grew fully within at 15, 20, 20, 10 alternative and all these within 3 weeks, and but failed to grow at 10 degrees C and 30 degrees C. So if the climate change will happen, either the cold spell will remain for a longer, or the climate will go a little bit higher end order 30, this embryo is not going to develop, and this seed is not going to germinate. So as the temperature warmer, this embryo will always remain or wait for the optimum temperature. That means it will change its season of the radical emergence. How we do this? We generally make a cut seeds, and we take the embryo length, either the binocular we use, or we use microscope, whatever, depending on the seed size. Then we use the ES ratio, embryo and seed ratio. If the embryo and seed ratio is less than 30, then we treat it, okay, this species can have high probability of the embryo dormancy. Now I'm going to second category of the dormancy, like a physiological dormancy. So low growth potential of embryo, difficult to overcome mechanical constraints of covering layer without dormancy breaking treatment. Means it is just like a lethargic embryo which is still very weak and not having that kind of the growth potential to push the seeds coat and come out due to some kind of the hormonal things, and that need to take a lot of time, that need to get some kind of the hormonal signal, sometimes or chemical signal, to grow and then push the seed coat. And found in various kind of the embryo, like 435 families of angiosperms, 61% of angiosperms out of 435 families, and all kind of the embryo, we have a different kind of the embryo. So in every 61% of the species have a physiological dormancy. And this physiological dormancy again has a two part. I'm not going to very deep, the regular PD or epicotyle PD. Then these have a different label, non-deep, intermediate deep, or non-deep or deep. Maybe you heard about the warm stratifications, cold stratifications, depending on how long you need to stratify it. If seed need to stratify it for only for the 2 week, maybe non-deep. If it require 2 to 4 week, it is an intermediate. If it require like a 6 to 9 week or 12 week or 16 week of the stratifications, then it's a deep. Means those species which is like, let's say, think about the Himalaya. Species are dispersing in autumn, and till the spring they remain inside the snow and they are cold stratifying. So those kind of the species are the deep dormancy. So ABA induced the dormancy, and gibberellin, light, cold and warm stratifications after drying or after ripening, or sometimes the nitric acid or butanoids, they break the dormancy. So what kind of the role these dormancy play? They fine tune to the environment, because the seed can germinate. If you disperse in, let's say, in September, you can germinate by the October or November. But what happened? That tiny seedlings, just look, visualize the picture of my daughter, baby, and then baby plant. If you have a first leaf, and all of a sudden the snowfall happen, frost happen, or cold wave will happen, the these first two or tiny leaves or very delicate leaf will die, then before the spring come, the majority of offspring will die. So that's why the dormancy break occurred during the season that is not favorable for the seedling growth. Thus, the full growing season is available for the seedling growth. So seed fine-tune the dormancy. Yeah. There is some questions?
No, sir.
Yeah. Okay. So it is fine-tuned to dormancy break such a way so the new seedlings get a full growth period. So by next winter or next hard season, they are fully developed and they can survive. So what are the conditions to habitat between the time of seed maturation, dispersal, and germinations? If seed disperse during the warm in habitat, dormancy break at high temperature. So seed dispersal, and then thereafter they're warm in the habitat warm. Like some seeds, if dispersed, like let's say in month of the June and July, then July, August, September are going to be warm. So they majority of them probability is that majority of optimum condition for germinations of the non-dormant seed made temperature lower than the dormancy breaking. So they will germinate little bit lower temperature. If some seeds are dispersing at a cold habitat, so they need a warmer temperature. If some seeds is going to be dispersed in autumn or winter, then they need to germinate in a warmer temperature, and they need the dormancy break need a low temperature. So depending on when your seed is going to be dispersed, you can give a warm temperature for the stratification or low temperature for the stratifications. So the PD is a reversible process. Other dormancy is not a reversible, but the PD is reversible. It is a cycling. So like a winter annuals and we have a summer annuals. What happened? Like they come out the break of winter annuals. During the winter annuals, they germinate and then they become a dormant, the seed. Then the next cohort of the seed will germinate. But the summer annual, they germinate at the summer temperature, they germinate at the winter or spring temperature. So PD is highly sensitive to environmental change, which diversity, that is subclass, label, and type of PD is a very complicated kind of the dormancy, have a different kind of the subclass, class, and label. Transitions between the PD and non-dormancy is regular because the physical dormancy, physiological dormancy, morphological dormancy, once it break cannot go back. But the physiological dormancy, when dormancy break, it can go back. So if the climate change will happen, that some drastic extreme climate will happen, so they can switch off and switch on. So PD and D is a transition. They fine-tune the seed stage of the life cycle to habitat conditions. What else does it mean? Then epicotyle dormancy. Sometimes you are not able to, because the seed are two kind. There is a two kind of the seed, that is we call the recalcitrant seeds and orthodox seed. Recalcitrant seed is always dispersed in a high moisture content, and if you dry them, they die immediately. And orthodox seeds are those seeds which can tolerate the low temperature. They you can dry them any low critical moisture content, and you can store them for hundreds of the years. Like majority of your agriculture seeds are a long-lived orthodox seeds. Majority of wild seeds or some of the wild seeds or tree seeds are a recalcitrant, like sal seed, Surya robusta. You cannot dry it. It lose the viability within a 2 week. Have you seen, majority of you already eaten maybe the lychee? With lychee or mango seeds, lychee seed, whatever the seed dispersed with the high moisture content, with the pulp, they are very short leaves. They die within, if you keep a mango lychee seed for the 1 week in just room temperature, they will just lose their moisture and they will die. But if you keep the moong dal or any fabaceae family, they will dry, but they will never die. So majority of those kind of the seed which is not able to remain viable condition in the seed, they have another kind of they germinate as soon as possible, but they remain in an epicotyle. So what happened? The radical come out soon, but the epicotyle take a time. In the meantime, it take a moisture content from the soil, and as soon as the temperature is or season is appropriate, they produce a first leaf and they start photosynthesis. So majority of these kind of seeds are bigger in size, like a mango, lychee. Similarly, happen with the oak seeds. These oak seeds disperse during autumn. Then after autumn, we have a little bit dry spell, and majority of these also have an epicotyle. Epicotyle means they germinate, the radical come out, but between the radical, if you the first radical come out, but from the radical to shoot, they take all night. The radical came within 20 days, but the shoot or the first leaf appear around 80 days. Similarly, the radical come out within 2 to 3 days, and it took almost 35 days. Similarly happened with the oak. So non-dormant versus dormant seed. Recalcitrance in nature. So majority of these big seeds with dispersing with a high moisture content are short-lived in nature, but instead of remaining the dormant, they have this epicotyle physiological dormancy, means their radical is coming out, because if radical is not come out soon, they will lose the moisture and they will die. So the radical is come out and radical penetrate in the soil, get a moisture from the soil and remain under the leaf. As soon as the enough season will come favorable, they produce a first leaf. So functions to minimize the risk of the seed desiccations. Now, look this, how we calculate these epicotyle dormancy. So once this radical come out, we keep these in these kind of the chamber and note down their date and keep them inside the incubator or the glass house or wherever you have a possible. Then these radicals come out. So you know, okay, this is the 0 days and how long they take from the radical to the cotyledon emergence. This is the Cardiocarinum seed. I done work in Japan. Now you got a flavor of the morphological dormancy, you got a flavor of the physiological dormancy. Now I'm talking about the morphological dormancy. Morphological dormancy means a combinations of morphology as well as a physiology. Sometimes the physiological dormancy break first, sometimes the morphological dormancy break second. So it depends on the species to species. So if there is an underdeveloped embryo, seed germinate very fast, has a morphological dormancy. If the underdeveloped embryo, slow germinations, the seed may have a morphophysiological dormancy. It's like that. Okay, I will come to that point. So now again, these have a different label: simple morphological dormancy and complex morphological dormancy. If something embryo growth happen at a warm temperature, we call it a simple morphological dormancy. If embryo growth is happening at a cold temperature, we call it is a complex kind of the morphophysiological dormancy. So this is the Cardiocarinum giganteum. Maybe in morning, Professor Rajsekharan is also showing this, talking about these seeds. So this is a Cardiocarinum giganteum, giant Himalayan lily. We collected it from the Uttarkasi, and then I bring its seed to Japan. So it's a flower in summer and it fruit in autumn. Then it disperses the seed in autumn. These are a capsule and these are like a fine paper dry seeds. So what happened? So we published this paper in Ecological Research in 2012, again with the Carroll and Jerry Waskins. And then I published another paper on morphophysiological dormancy or epicotyle dormancy on viburnum with the Jeffrey Wallach and Sethi, who published that global change biology paper. And then we again published the paper on Convallaria kissicki on morphophysiological dormancy. What do you mean by morphological dormancy? It's like that. A baby, I always, my teaching, I always use the parallel, the scene. It's like a baby born premature. Like if you look here, this seed is dispersed from the mother plant. It's like a seed is dispersed from the mother plant. It's like a premature baby come out within a 7 month. Then what we do, the doctor put it in an incubator, give all kind of the nutritions, and by when the baby remain in an incubator for a 3 months, around 3 months, until I complete the 9 month, and then in the meantime all the organ got developed. It is like that. It dispersed from the mother, something like a premature. It is 19 December 2016. By September 2007, it remained like nothing happened. Then it start developing October onwards. November, within October to November, it developed fully. Then January, February, March, April. So 2006, seed detached from the mother, and by 2008, it start germinating. And how we done this? This is again a photograph from that particular paper. We kept these seeds under the snow like that. Every month I'm used to dig one bag of the seeds, cut them, look the embryo size. So every month I'm taking 10 seeds, cutting these embryo, measuring this embryo size, and looking for this happen almost for the 2 years. Now, when I try to, see it with the climate change or the temperature. So this is the embryo development. This is a radical emergence. This is a cotyledon emergence. So if you look, the embryo start developing after the one winter, and this is the spring, then summer, then autumn, and then embryo start developing. Then the second winter, the radicle start emerging, but the next spring the cotyledons come out. Then we look, it's the same in a laboratory. We use the artificial temperature. This is like 25-degree day temperature, 15-degree night temperature. So this is summer temperature, this is autumn temperature, 15 and 5. Then winter temperature is 0. Then again summer temperature. Somehow we are able to develop these seeds within 1 year, 360 days. But in nature, it took from 2006 to the June 2000, it almost 18 to 20 months. Somehow we managed to grow it in 12 months, but still now how these temperature play role. Generally what we do, we keep the seeds inside the incubator and check them for the month or 2, and it's okay, they are dead, they are not dormant. No, to break a dormancy, you need this kind of the sequence. You need to simulate the nature and look if climate change is going to be happen, if extreme weather going to be happen, if this period is going to be shorter or longer, or there is some interruption in these periods, the seed is not going to germinate. This is the lily of the valley. I like this plant. This is one of the horticultural species, especially famous in a northern hemisphere. So we done that same work with this. In this case, the embryo radicle emerged first, then embryo developed later stage, and radicle come out in January 2009, and the cotyledons come in May 2010. If someone is working on a seed germinations and talking about the seed germinated based on only the radicle, so it is a half story. So always look how long from the radicle to the cotyledon, because the cotyledon emergence is depend on the what kind of the temperature or climate the seed is going to get during that particular period of time. This is the seed, the embryo developed. In this case, embryo, instead of developing or longitudinal, it developed more with the width, not with the length. So like here, the length remains same, but it start like swelling. So this way, this is the, now I will talk the physical dormancy. Majority of we Indians work on a physical dormancy. It is much easy to break. Whatever the seed had a high, the hard seed coat, we always give them or acid escarification, boiling water without knowing either the seed is going to imbibe the water or not. So physical dormancy, cause of the water impermeable seed or fruit coat. So what happened? Every seed has some kind of the water gap. If the seed is not able to imbibe the water due to the hard seed coat doesn't mean that akhrot, the walnut has a hard seed coat, but the walnut can imbibe the water from these cracks. But on the other hand, the beans or some of the chana and other, they may have a hard seed coat, or majority of the Fabaceae species, acacia, albigia, all these kind of the Fabaceae, they have hard seed coat. They're not going to imbibe the water. So they have the water gap that act as a environmental signal detectors from the, okay, if you are working for the nursery, you are working as a horticulturist, you can break these hard seed coat by the escarification to have a cut, or to put them in a just dip in boiling water or dip in them in acid. But what happened in nature? Nature is not good. No one going to in nature to make a scar on that, to cut the seeds. They have some kind of these water gaps. These water gaps are very sensitive for the temperature and relative humidity. If they get a proper temperature or a proper humidity that is a species specific, then these water gaps open and get the water. If that climate or the microclimate is going to be changed a little bit, this process will not happen and these species will not able to germinate. What is their natural season? They may germinate, they may come out of the dormancy, the beyond their natural seasons. So the unplugging of the water gap in the seed or fruit coat is not a random event. And the timing of germination in seed with the PY or physical dormancy is environmentally controlled in nature. That's why I say seed ecology is going, like those who are in a biochemistry, or Arabidopsis is a model plant for any kind of the molecular biochemical analysis. Similarly, I say seed is a model tool to study the climate change. If anyone want to work on a experimental base, policy label, looking the climate change data from the remote sensing and other things, that is good. But we need all evidence at a micro level. What's going to happen? Not only the adult plant. People say, okay, leaf are drying, flowering is happening early, or the growth period is happening. Well, we know it's happening, but how that regeneration process is going to happen, we need to learn a lot about at the level of the seeds. Now, if you go to the micros, this is again my friend from the student of the Baskin and Nalin from Sri Lanka. He worked on the geranium seeds, and these are the pictures of these water gaps. How these water gaps have within a 2 minute, within an 8 minute, within 12 minutes and 20 minutes, they get ruptured if they get an optimum temperature and relative humidity. And some seeds, these have the pleurograms. Maybe you notice acacias and albigia seeds, these pleurograms also play. If the seeds have not a water gap, they do not otherwise have a water gap, and these pleurogram is trying to get a water, and they play a role to imbibe the water in nature. How we do it? As I said, any hard seed doesn't mean that that is a water impermeable. So we need to take a seed coat is a very hard, fibrous, difficult to scarify. You can use the methyl blue to ensure the seed is permeable. So we put a seed like in petri dish, and we take their dry weight, we keep them on a wet surface, then every 1 hour we just blotted dry them and weigh them and take, okay, how they took a seed water. So this is the non-scarified seed, means we not make any kind of the cut. This is again with my paper with the Baskin on Dadunia viscosa. Then we dip them in a boiling water, but just for a 5 second, nothing happened. But when we mechanically scarified, we make a cut. So within a 24-hour, these seeds are not able to imbibe the water, but this seed able to imbibe the water. That's so the seed has a physical dormancy, means seed coat is impermeable. This physical dormancy also in nature, it break either the fluctuating temperature, because like if you look these Rajasthan Churu or Mount Tavu, you have a like night temperature is minus, and then during summer they have a high temperature. So these lot of temperature fluctuations make a crack on the seeds, and that help the seed to germinate, to get any kind of the crack, and these cracks help to enter the water insides. So low and hot temperature play a role. Sometimes the fire, like in Banksia, fire is play a role to break up PY. Like here they put some Fabaceae seeds and they make a fire, the seedlings are germinated, but in this case they not make any kind of the fire, the seedlings are not going to be germinate. So the fire also play a role to break a physical dormancy. So fire is not always a bad, fire is a good tool for the ecological point of view. And if you are doing restoration ecology, sometimes these tractors and the machines also make some kind of the cracks on the seeds, and the seeds get the hard coat get damaged and they germinate. Now I'm coming to the last, combinational dormancy. Combinational dormancy is going to be a physical plus physiological, means first the seed coat has a impermeable layer, and then once it break, then it embryo has some weakness or some low potential growth, so it is not coming. So it is a combination of the physical plus physiological. So it happened PY plus PD is evident in seed with the water impermeable seed coat combined with the physiological embryo dormancy. So depending on the species, they lose PD components occur before or after the PY. Now, if you look the world biogeography of the seed dormancy, world distributions have a 5 class of seed dormancy. If you look the majority of subtropical and tropical zone, if you look these different zones, the PD is one of the most dominating. Then we have a PY. If you look temperate, again the PD is one of the most dominating kind of the dormancy. If you look the 535 families of the angiosperm, 61% have a PD, 20% have a morphological dormancy or morphophysiological dormancy. Only 4% has a physical dormancy, and 2.3 has a non-dormant. So only 2% seed in the world has a non-dormant so far, the based on the study. So seed, majority of seed has some kind of the dormancy. But please, whenever you give any kind of the treatment to the seeds, first look the phylogeny, first look which species that belong. If some species not belong to the family known for the physical dormancy, you're putting them in a hot water or acid, you are going to damage their embryo. Don't do that. So this is the scenario. PD is most dominating, then PY, then MPD, then MD, and then PY plus PD, like that. So how to break the dormancy? Now you are having a lot of questions. If the seed has a morphological dormancy, give after ripening time in incubations at an optimum temperature. You keep them in an optimum temperature, just keep eyes on the embryo. You need keep some extra seeds. Look, either the embryo, you can take embryo length just as a fresh seed, and when the radicle is about to emerge, then take an embryo. If the embryo is developed inside, then that has a morphological dormancy. give a stratifications, either the warm or cold stratifications, means keep the seed in moist condition in a low temperature or a warm temperature, or keep them on a laboratory desk or a room temperature for after ripening. That means they have a PD. If the MPD, morphophysiological dormancy, means morphological as well as morphophysiological dormancy, apart from giving them a stratifications, better to keep them in a sequence of the temperature, autumn temperature like 90 days, depending on the where you are living. If you are in extreme north in a northern pole, then winter is maybe longer. Winter maybe 120 days, winter maybe 150 days, depending on the 90 days. Autumn 90 days, winter 90 days, spring 60 days, summer 120 days. Like we simulate these things, and then we keep at some point of time the seed will start germinating. If the seed has a water or physical dormancy, either scarify it or keep them high and low temperature exposure. If the seed has these two kind, then you have a scarification and stratifications. Now I again come to the climate change. Now you have a good understanding of the dormancy and germinations. Now I come to the climate conditions again. So we know to break a dormancy has some species has a narrow, some has a intermediate, some has a wide. In current scenario, this is the label of the solid line and a duration. This is a solid line, which is a label, and this is the durations of a cold stratification changing currently and will likely to continue to do so in future. So that's this is the current scenario. Some need a low temperature for a short period. Some need a low temperature for a intermediate period. Some need a low temperature for a longer period. What happened? In future, this temperature is going to change outside the season. Maybe they require the low temperature at this particular point of time, but it is going to happen a little bit later. So in this case, the decreasing effect of the warming, so what happened? The dormancy is not going to be break. So it is going to hamper the plant regenerations. And then another label, like climate change affect the temperature and moisture, seed maturations and mass. So seed dispersal, if weather will change, the maturation time will change, the dispersal time will change. Once the dispersal time will change, it affects the dormancy, it affects the seed germination, it affects the seedling emergence, and it also affects the input of the seed in a soil seed bank. And then overall the seedling survival and seedling growth will get impacted, and it is going to change the population size, structure, and desynchronize life cycle events, modify competitive interactions after the distribution range of various species and types. Like if majority of the Indian species, they disperse their seed during just before the monsoon or during the monsoon, thinking that monsoon will arrive in third week or fourth week of the June in Delhi or in North, they disperse their seed just a week before. If the monsoon get delayed, these seeds will going to be dry and the seed is going to die, and that is going to be a catastrophe for the plant population and plant regenerations. So climate change, to understand the climate change, we need to understand the regeneration ecology of the wild plant. Agriculture, we know everything, we can manipulate. Now, if you look the work, what kind of the work? Studies related to the climate change. Tundra, lot of the studies happen. Boreal forests, because they are part of the European and North. But if you look our part, like tropical forests, savannas, and other temperate region, we have no seed study related to the climate change. A lot of work need to be done. And based on this is again from that paper, they set in 2019, they set a research agenda for seed trait functional ecology. If we want to understand, because we talk about I often call it a plant blindness. When you go to the funding agency, you want a tiger biologist, you want an elephant biologist, you want a bird ecologist, the DST is going to give you fund. But when you talk about them working in seed ecology, they treat you just like a nurseryman. Oh, you are a nurseryman, you are a forester, you can. No, seed ecology is going to be much, much more. And majority of these authors, including my professor and then Jeffrey Wallach, and they are majority of my good friend, we are now developing a handbook for the seed traits. They set this paper in 2019, paper published, research agenda of the seed fruit functional ecology, seed traits, and it is already has more than 60 citations within a 2 year, despite of the corona. So the take home message from my this talk is more studies on plant regenerations from seeds are needed, particularly from South Asia and other global South countries. I am reviewing paper, I'm editing journals, and unfortunately, we are not getting a quality seed research from, especially from India. I have no doubt. I'm sometimes feel regret. I don't have my own group. So we need a much more. India is have a lot of biodiversity. If you talk about the medicinal plant, Rajshekharan was talking about the export quality of these medicinal plants. How long you extract the medicinal plant, the raw material of the medicinal plant from the nature? You need to look of their regeneration ecology, and you need to domesticate them. To domesticate them, you need to know their seed ecology or their seed biology. That's all from my side. I think I'm on time. We have a question.
Yeah, yes, sir. You are absolutely on time. And now the session is the open for the and discussion for the question and answer. So any query or any doubt or anything you can ask to the sir.
I will be happy to interact with more with the students, because
Yeah,
in fact, whenever in my if anyone is interested in seed ecology, I use my Twitter handle only for my scientific work purpose. On the first slide, I put like @Syam Partyal. I generally post the seed ecological step, plant regeneration ecological steps, as well as some kind of the PhD and postdoc kind of the opening, and sometimes the master level fellowship opening. So if you are interested, you can find a lot of my own work or my colleagues' work or the work from the all around the globe. And I always encourage, whenever I find any Indians or any South Asians working on this field, I always try to share his or her work in my Twitter handle. And majority of my Twitter handle, we have a 90%, more than 90% are ecologists only. Yeah. Go, ma'am.
yeah, sure. Kalyani ma'am, you can ask the question.
Okay, thank you. 1 second, I just have to do some little bit. I'm audible, right?
Yeah.
Yes, yes.
Yeah. Dr. Fartial, I think this was an amazing talk. And we teach seed dormancy as a part of a physiology course.
Yeah.
But I never anticipated this kind of a twist to the the theoretical aspects the way you have given. So I think I must congratulate you on that. And also less of a question, again more of a comment I would like to give, a compliment rather, is that seed dormancy is generally taken in view of the cultivated crops, while you have shifted your attention to the wild plants, which is commendable. Because if we have to like one example you gave is of medicinal plants, but I think in this decade of ecosystem restoration, I think this work is of absolute significance, because if you have to restore an ecosystem vis-a-vis climate change, then it is the wild plants we are supposed to look at, and not just the cultivated ones. So I think if you manage to get students, I mean, I'm sure lots of students would be inspired to join you now, and you can probably work along along the lines in rest.
Yeah. Yeah.
I wanted to congratulate you on this, the kind of work that you are doing. Thank you so much for telling us.
Just if I permit to share my screen. Can you see my screen?
Yeah.
Yeah.
No, sir. No. You can again share, sir.
Okay. I'm going to share my screen. Okay. Just look. So, ma'am, I not only work on the wild. I work from wild plant from wetland to the woodland. So, look, this paper recently published, like a sweet and musky scent of home, biogenic ethylene, fine-tuned seed germination in wetlands. And if you look this paper, seed germination in mudflood species responded differently to prior exposure to the hypoxia. And ready to change seed traits contributions and high adaptability mudflood species, seed germination community shape along the hydroperiod gradient. So I work from the tree species to the wetland to the mudflood to the savanna species. I never work in agriculture, and I keep my focus only on the wild seeds, and love to have this kind of the more interactions, and always ready to open if anyone because sometimes it is not I'm not, if some of your students want to work in these aspects in future, I always be ready to help whatever the capacity. If you want to me as a advisory board, as a co-supervisor, any kind of the things, I always be ready to come forward.
Yes,
Yeah,
thank
sir.
you so much. That's what was
Sir,
expected. Thank you so much.
because it will be very big, because one question is the my side also, as you are the given to the idea of the co-supervisor or the research work. So minimum, because the student is the basically approaches to me for the only for the dissertation purposes.
Yeah.
So it will be the 6 months to 9 months. So in this period we can do some experiment or not in this.
Yeah. Even what I do, I always say you cannot eat the whole watermelon in one go. To eat a watermelon, you have to make it as a piece. So I take a digitations or a small component, and then like a 3-4 digitations can make a good paper. So you
Okay.
take identify one problem and give a one component of a digitations. Let's say you take a one species of from Delhi, and it's okay. This few experiment one student can do, few experiment second student can do, or you can do it like that. So it can be done. I always in favor, because no, if I want to finish in watermelon, I cannot eat a whole watermelon in one go. So I need to make it a piece, and then to do a good research, you need to do it in a pieces.
Okay, sir. Thank you so much, sir.
One piece we will take it from VIT also.
Thank you, my dear. Raja is one of my oldest known figure in this, because we are a good friends of our PhDs.
We are good, not only good friends, we used to call brothers. In such a way, in our long association since 1998. He has seed biology, seed germination studies with various mallotus species and all. It's a rememberable journey, and he become Humboldtian also. That's a great thing. And yeah, but I would like to have his involvement with our society as well as with our student community. That's a need of the hour, and his expertise we should make use of it. I give an appeal to all our students. Please do catch this Humboldtian. Don't leave him. That is message I would like to pass on.
I will love to, if someone can become Humboldtian after my writing a reference letter.
Yes, good. That's the thing we need. We need such a interesting the openness in the open forum to say this one. giving a consent or nominating on fellowship. We are struggling a lot, but fortunate on this forum he has given an open statement. Please do catch him and do good work, and I should find much more Ambolians in days to come.
Thank you, Raja.
Okay.
Yeah, now thank you, sir. As in the beginning of the lecture, I said that no one can explain this seed ecology better than you. So all the participants is happy, and I can say really it is good for us. So thank you so much, sir, and you are sparing time for us for the one invitation. So I am really thanks to you.
It's always pleasure to talk with the students and the colleagues. Thank you.
Okay, thank you, sir. Now, after the first talk in the second session, we are moving to the second talk, and second talk is the Professor R.P. Singh from Lucknow. So for the introduction of our second speaker, I invite Dr. C. Rajasekharan to introduce to our next speaker.
Hello.
Yeah.
Yes, sir is available.
Yes, sir.
Sir is available. Sir, good afternoon, sir. Hello. Sir is available on our this thing.
Yes, sir, is he available?
Sir.
Yeah, I think there might be some connectivity issues because it's just 3 o'clock. He asked me how many after the some time it will be happening. So I said that 15 minutes.
Or otherwise, just give it to him so that he will be because he is log into our this thing. Just to give a ring to him.
No, he is already logged. I can just again call to him.
Log in, and I hope.
Yeah, just a moment. I will call. Yeah, just a moment, sir. Is he coming to this in front of the system? Yeah. Yeah. So now, yes, sir. So yeah, welcome, sir. Now I request to the Professor C. Rajasekharan to introduce to our next speaker, Professor R.P. Singh, sir.
Yes. Good afternoon to one all. It's my immense pleasure to introduce our teacher, Professor R.P. Singh. First time I met him at New Delhi only at IRA or the eve of Second International Congress on Plant Physiology. Before that, sir has revised my paper in helping in publishing Physiology, Molecular Biology of Plants during 1998. That's the first occasion I happened to get interaction with Professor R.P. Singh. And then subsequently met at 2003. Since that, he is a mentor to me also, and he is having great the contribution in the field of our academic and research activities. He started his career from Rohtak University, and first he did his PhD from abroad, then PDF with Canada. Then he began his career with Maharishi Arvind Dayanand University, Rohtak. Then he joined with the Bhimram Ambedkar Central University, Lucknow, somewhere 2005. Since that, he is not only serving as a professor, various academic capacities. Almost all the academic, administrative positions he hold it. Yeah, right now he is serving as a rector to this institution also. Under his research, contributions are at par with his academic and administrative activities. He has guided successfully to 32 PhD scholars and hundreds and hundreds, more than 100 MPhil and PhD, MSc dissertations also. He visited various countries with various schemes. Almost 20 plus odd countries he has visited, and also he has represented our Indian subcontinent at UNESCO this thing. And publications, when you take that is more than 200 publications he is having it, and authored many publications, many books. One of his book only, when I started my PhD, nitrogen nutrition in higher plants is a first book to refer also. That's he has written along with our Professor H.S. Sriperumbudur also. Is one of the very important, or otherwise I can say Bible for our nitrogen assimilation studies. He is having a very good gravity on this field. And again, when you talk about the editorial activity, this thing, since 1998, I'm finding as a associate editor and editor to plant physiology, molecular biology of plants. It's one of the hallmark journals in India, which is having an impact factor around 3. It will be shooting up also. Sir is having very vast experience in this field. Few of them only I'm mentioning here. Apart from that, he is a fellow in various national and international bodies. Likewise, his contribution in this field is indefinite. That's alpha. We used to say botanically this one indefinite. Likewise, number is high, and again his curiosity on setting up of federation of our biological societies is vision, and also doing service to the society is fashion. With these words, I would like to request our professor to deliver his brainstorming lecture to our participants. Thank you very much.
Thank you, sir. Thank you, Professor Rajsekaran. It was so nice to hear you, but you just gave so many things about me. Thank you very much. I am obliged. Dr. Abhishek, Dr. Rajsekaran, and so many other colleagues who are there in the conference, the participants from various colleges and universities. Today, I just want to share with you some of our ideas and some of our work related to a topic which is not very widely discussed. That is designing novel ecosystem in Westland. So the topic today I have selected based on some of our recent study, and it fascinates me very well. Means I am very fond of this topic, because I feel that this is time where we should think of these kind of approaches. We are talking about climate change, and we are facing about climate change and global warming, and many complex changes are going on throughout the world, and we are possibly not prepared that way to meet the challenges. United Nations is also discussing on various aspects, and it has presented its goals recently. Amongst various goals posed by United Nations, sustainable development goal, like sustainable development goals Goal 2 has a focus on finishing hunger to achieve food security and to improve nutrition to all through the sustainable agricultural practices. The benefit here, I would like to say that on one hand, there are decrease in land area, there are degradation in agro ecosystem and all kind of ecosystems due to intense man-made activities, and then there are requirement of uh, production of food with diverse nutritional value. And then one major challenge which is coming in agriculture is decreasing interest of producers in agriculture sector. Farmers, traditional farmers are leaving. The land areas are decreasing, generation by generation. Climate change has posed new kind of threat to the agriculture, and simultaneously the soil fertility has decreased. Chemical contaminations has been very serious in entire ecosystem and our food bowl, and so on. So under these challenges, we have to plan some innovative approaches to meet out the mitigation requirements of climate change and to feed our mounting population the food which is grown in sustainable way, and ecological way is sustainable way. We all have to believe it. Non-contaminated food, nutritional food, it can be achieved only when there will be diversity in our food bowl. So with all these things, we can achieve sustainable agriculture. And here, in such circumstances, we have to think in some novel way. The beneficial soil microbes, crop diversity, and adoption of ecological and economic principles can be significant tool for achieving it. In addition, mitigation of climate change through carbon sequestration by biosphere can be a great deal for carbon trading, which is coming as a very significant new economic system in the world. If we look on the problems of degradation, there are just this data indicate the fractional forest cover map of South Asia. You can just see throughout the world the forest cover is declining, or it is fragmented, or it is very, very less than required. But South Asia especially is the area where we live is suffering from the problems related to decline in forest cover, and we have a scope to increase it. Desertification or land degradation is a reality. I have just kept the concern of India here in this slide, and you can just see that throughout the country, in all kind of ecosystems, various type of degradations are there, and this especially this data released by the government, it indicate very clearly that during 2003 to 5 and during 2011 to 13, between this the data has two periods, very, very small changes has been occurred in degraded areas in India. In all arid, semi-arid, humid, all agroclimatic conditions. If you look on vegetation degradation, water erosion, wind erosion, salinity, alkalinity, waterlogging, mass movement, frost shattering, man-made causes, barren land, rocky land, settlements, there are not much differences in the total, say, degraded areas. So, say, over a decade or so, we could not achieve, and if we go with, I could not find the more recent data, but if we shall go for that, we will see that not much difference is there. Our progress is, the point I just want to underline, that our progress is not meeting out the requirement which has been, say, posed more seriously after this realization of climate change and global warming, increasing disaster, increasing population, all that. So we have to think in other way. The pace we have taken may not be sufficient. Well, there are a lot of challenges in ecosystem restoration throughout the world and in India too. You will see that United Nations have declared a decade of ecosystem restoration as theme for International Environmental Day to underline the requirement of ecosystem restoration. And it has been mentioned that there should be reimagination, recreation, and re-establishment of ecosystem as a principle of ecosystem restoration. We have in India, and almost all countries have their own, their own ecological and economic policies and regulatory rules and schemes or programs for their development. We have many. You just see, for ecosystem restoration and biodiversity conservation, we have so many national ecological policies and national socio-economic policies. Afforestation policy, agroforestry policy, biofuel policy, Clean Ganga mission, National Water Mission. These are very major national ecological policies. If you look on national socio-economic policies, they are rural tourism, housing for all policy, guaranteed rural job policy, doubling farmer income, and obviously it will require several kind of capital stock and ecosystem as input, and then there will be novel outcome if we shall be able to go for significant achievement in ecosystem restoration and biodiversity conservation. I am just going to share with you some of our ideas and some of our work in relation to ecosystem restoration and biodiversity conservation, which can be one way to do it. This is designing novel ecosystem in wasteland or barren land. What will be this novel ecosystem and why it will be required to be designed? Why to design a novel ecosystem? Restoring degraded we are talking about restoration of ecosystem, but I believe that restoring degraded ecosystems may not be possible as we don't know about the past below ground and above ground biodiversities and abiotic components which have been changing time to time due to changes in various agro-aqua climatic conditions. different time there are different kind of biodiversity. Each rather if you go by decade and decade and decade, there has been significant changes in agroclimatic or aquaclimatic conditions. There has been changes in biological components. There has been changes in their interactions, relations. There has been changes in their say relations with abiotic components of ecosystem. We don't know all that. We don't have record of all changes at micro level. We know, but something is there in hand. We know that a good number of beneficial soil microbes which can help the plant productivity and carbon sequestration by providing nutrients, releasing hormones, controlling pests, degrading organic nutrients which are toxic more rapidly, and remediating toxic contaminants in soil and water are possible. Its novel consortium can be engineered in plants atmosphere. This thing we know, and a lot of science is going on studies related to microbes and their uses. Many beneficial, economic and high value plant and animal combinations can be designed and arranged with various layout and landscaping designs for multiple economic and ecological benefits, which are suitable for different agroclimatic, aquaclimatic, and socio-cultural solutions. So this way, even if we do not know what was there in our previous non-degraded ecosystem, if we are planning to restore, we can design below ground diversity and above ground diversity as per the emerging need, as per the emerging agroclimatic or aquaclimatic conditions. And this is why the restoration, ecological restoration, has been considered to reimagine. We have to imagine what is required now. We have to imagine what would have been there in that kind of ecosystem during previous time. So we can imagine, we can say recreate the situations, and then we can re-establish ecosystems of our various ecological, cultural, and economic uses. So this will be required for restoration, and that's why we require. So that we shall require, and that is basically the redesigning of novel ecosystems. There are a lot of benefits of the design novel ecosystems over the native biodiversity, wasteland, barren land, and degraded wetlands. Here I have just put some. You can just see, I will not read all, but you can see that there are certain problems with all these kind of, even if you just look on native biodiversity, there are extirpation of selective communities. Many communities are losing their potential, are losing their identity by various known and unknown reasons. Loss of genetic diversity is there, and underutilization of soil resources are there. We cannot afford with increasing population and decreasing land resources. We cannot, means it is said that basically productivity per unit area should be increased, but there is a limitation of bearing capacity of nature, and we cannot increase artificially the productivity beyond a point. So there should be other ways also to enhance the ecological and economic benefit of the ecosystem, and that can be designed, and through that we can achieve many beneficial outcome. Even in westland and riparian belts, that is problem, certainly barren are degraded areas are problem, have additional greenhouse gas emission source, invasion of exotic weeds, unwanted, if you leave it unwanted, more resilient uh, plants, animals, and microbes will invade, and erosion of nutrient leaching will be there, but we can convert it in sink. We can redesign the composition of endemic species and go for ecological restoration and better nutrient recycling and soil stability of that system. Barren lands can also be redesigned on the ecological and scientific principles, and degraded wetlands even can be redesigned. Some urban designer ecosystems are here throughout the world. These are examples of some urban designs, which are ecological designs, considered as ecological designs. Some specifically designed ecosystems for bioremediation have been shown on the screen. You can just see specific beds of plants and microbes are designed, and they are subjected to treatment of water. This is one very considered very cheap and very aesthetic way to remediate the degraded ecosystems. This is one example. There are so many examples where the microbes and plants have been used to remediate the degraded water, contaminated water. If you look on the forest ecosystem, there are monoculture, and that also get degraded at one time because of economic harvest. And due to so many regions, the various pockets in forest areas have a very serious degradation. So that has to be redesigned. Even new forest can be redesigned, and new, the pockets, degraded pockets in the forest can be redesigned on the basis of the designer ecosystems. In Delhi, there is, many of you might have visited, there is a biodiversity park which has been developed in collaboration with Delhi University, especially Professor C.R. Babu led project, and Delhi Development Authority for multiple purposes: native species conservation, carbon sequestration, bioremediation, and multiple ecosystem services. So these are various kind of, I just tried to give you an idea of various kinds of this designed ecosystem throughout the world. It can help. We have published this idea, and it has we have just underlined that designer ecosystems can be a solution for the conservation exploitation dilemma, because natural resources are required to be utilized also. There are exploitation of natural resources, and then there is need of conservation. So we can design pockets for different pockets for conservation and exploitation, and this way we can get help of this whole concept. You can just see the riparian belts that may be from the pictures, maybe from different countries, but you can just see the difference in its ecology, its nutrient recycling capability, its its aesthetic value, aesthetic look, and its multiple ecological and economic benefits. So why not we can redesign the one which is on left, not exactly the same, but similar kind of ecosystems can be designed on what has been kept on the degraded part. There can be so many multiple benefits of economic and ecological benefits of ecosystem if it is designed in riparian belts. Say it can decrease erosion, improve water quality, restore ecosystem, maintain biodiversity, maintain river courses, decrease insect pest, decrease weedy algal growth, and maintain fish stock. It can increase capital value of the land or water, shelter effect can be obtained, retention of nutrients can be there, lowered water table will be there, denitrification can be obtained through these designing, opportunities for diversification, recreation, cultural and spiritual fulfillment, and ecotourism. See, what we want out of that degraded ecosystem, the whole regeneration philosophy will depend on that. The combinations of microbes, plants, and animals will be accordingly. Then there is a lot of conflict on bioenergy, bioenergy crops and food crops. So we have proposed that perennial cropping system for bioenergy production can be done on marginal land, and a lot of marginal land is lying where the new kind of perennial cropping system can be generated, and once the ecosystem will become suitable for cultivation of food crops, then food crops can also be cultivated in those areas. So this can be a way of restoration. You can just see in any kind of degraded say soil or agroclimatic conditions, there can be very huge biomass of perennial grasses, which are very diverse and very climate resilient. So they can be planted properly, and we have done some systematic studies also. This is a field study, and then we have done some experimental studies on this, which I will show you. Say microbes, many of you might be aware about multiple role of microbes, beneficial soil microbes especially, and they are seen as one very important tool for agricultural sustainability and nutritional improvement. There are multiple evidences which show that microbes and plants might have co-evolved. Even some animals, microbes might have co-evolved with animals also. So metagenomic studies reveal that about 99% microbes are not culturable on the culture media in practice. Hence, we know only about 1% say beneficial soil microbes, which are characterized in laboratory studies, and only a few formulations are commercially successful. There can be various kinds of role which can be played by microbes in agroecosystem after inoculation or after regeneration, and this is just promote plant growth and growth by growth hormones. It can act nutrient facilitator by siderophore and chelator production, convert non-phytoavailable nutrient into phytoavailable from the solubilization, act as biocontrol agents against various plant pathogens. It can degrade toxic organic agrochemicals or its residues. It can support plant growth and yield under the abiotic stresses. It can help in atmospheric nitrogen fixation, and it can increase the surface area of fruits. So many these kind of benefits has been reported. So but see, there are a huge knowledge gap and bottlenecks in large scale adaptation of bioinoculants, plant growth promoting rhizobacteria, or plant growth promoting microbes, which are it's a very popular name in agriculture. This is require attention if we want to achieve the good benefit of these microbes in restoration, ecological restoration, and agriculture, rather production of plants and crops. Long-term field trials of multiple years on multiple locations are very rare. Adequate knowledge on competition in inoculated PGPMs with native soil microbes and other soil organisms and its ecological succession in different agroecosystems are hardly available. Adequate knowledge on impact of agroclimatic conditions and climate resilience of inoculated PGPRs in crop field are rarely available. Effective mechanisms for marketing management of bioinoculants up to the end users in most of the commercialized products are poorly established in most of the countries of South Asia. Lack of adequate knowledge on promotional policies and regulatory provisions of governments in relation to bioinoculants for its production by small-scale entrepreneurs and its use by the farmers, which could have motivated them to go for it. Lack of adequate knowledge on toxicity potential of synthetic fertilizers, pesticides, and other agrochemicals to the end user is there. So these are certain very pertinent knowledge gap and bottlenecks. In addition to these inoculated microbes, there are a lot of endophytes, and these endophytes are microbes which are considered to be co-evolved, or they have infected them later and they are existing with plants in a say symbiotic mode. So we know many of them, they can be of various use, bacterial endophytes, and there can be a lot of examples are there on bacterial endophytes which are increasing the plant growth. And there are fungal endophytes which have been reported to enhance the plant growth. These are certain examples, but there can be many more. Then the third aspect is whatever beneficial native microbes are there, they are also required to be regenerated. And they can be regenerated only when the ecosystem, soil conditions, and the the rhizospheric nick will be restored, because they require certain level of organic organic matter in soil and non-toxic conditions, and also the high temperature or further abiotic stresses also affect them. And in addition, they require some associated plant rhizospheric nick for their rapid growth and multiplication. So if it is said that conservation agriculture is one which is suitable for this, so we have to see through which kind of approaches we can regenerate them. Low external, there are so many kind of environmental damage and human impacts which have degraded the entire soil microbial ecology, and either their population has decreased or their density of those populations which are there have decreased, and the threshold level of those microbes are not there. So we have to regenerate or re-inoculate them, and then we have to make use of them for benefit of plant and benefit of sustainability of soil fertility. So low external input agricultural practices can regenerate. Increase in soil carbon pool can regenerate, help in regeneration. Maintaining soil moisture to optimum by drip irrigation or mulching can help. Conservation agriculture practices can help. Organic agriculture can help. This zero budget farming is one popular technique in which it is some efforts are made to multiply them, and then rhizoengineering of microbial bioinoculates can also regenerate the microbes. This I have just mentioned because we have experienced, we have studied that even it is not only native, exotic microbes also work very well. If we look on green revolution, which are related to agriculture and plant productivity, there have been certain claimed advantages of it, and it benefited the whole world, India at a time, but now a lot of claimed disadvantages are there. And we know that it has resulted agroecosystem damage, food contamination, loss of soil fertility, and decline in water table. And it has increased input cost, borrowing, and losses to farmer in disasters and epidemics. It has promoted monoculture, super weeds, and super pests. So with all these things, basically, it is considered unsustainable now for production system, maybe plant production system. Consumption of chemical pesticides in India and in whole world has been a very serious concern, and it is not decreasing. We have recently reported that there are severe contamination of carcinogenic heavy metals and metalloid in agroecosystem, and their associated health risk has been found very, very dangerous. Here you can just see the toxic metals from various markets, the vegetables, different vegetables taken from ready to use vegetables taken from various markets of Lucknow, and they have been found very having very high total target hazard coefficient. Here it has been found that nitrate, fluoride, iron, salinity, arsenic, lead, chromium, cadmium, etc. in are at worrying level. And in this report, it has been said that 18 districts of India are having very worry causing level of these toxic substances in food system. Lot of reports in papers appear and goes, we all know it. So we are having poison in our food bowl, and we are not that much concerned. We should have been concerned about that. Unassimilated nitrate, nitrite, ammonium, and phosphate are there, which are of fertilizer origin. Heavy metals from fertilizers, pesticides, weedicides, and contaminated irrigation water are there. Toxin of microbial origin from pathogenic microbes are there. Arsenic of geological origin through groundwater irrigation is there. And contaminants from water, milk, cold drink, fruit drink, and other liquid intake are there. So microbial agents on fungal pathogens can work, and even in place of pesticide, we can go for various kinds of biocontrol agents, and trichoderma is one which has been proved very significant for sustainability of agriculture, and there are many biocontrol agents which can be used. We have just used certain in tomato. This is our own work, and then we have to shift from organic principle, rather towards organic ways, ecological ways, which can provide. We have to go for innovative ways which can provide higher productivity without chemical inputs, and it is a major challenge in non-chemical agriculture, essential for agricultural sustainability and non-poisonous food security. So our focus has to be changed from increased productivity alone to holistic integration of natural resource management with food and nutritional security. So organic principles may contribute to a valuable framework for future sustainable agriculture production. Hence, innovations and technological inputs are essential. We have isolated some exotic microbes from, we have been looking for climate resilient microbes, and for that we just went to a plateau in Maharashtra. So we have been looking from different harsh ecosystems, and from this ecosystem we isolated certain, we studied the microbes, and we just found that in rock there are possibility of having microbes, and by this genetic analysis we found a very huge number of fungal and bacterial genera, about 256 genera were there in the samples, and we got them identified, and finally we isolated some 21 fungal and bacterial genera from that ecosystem, and we could study their various properties, especially PGPR properties, and simultaneously these are PGPR properties and these are various abiotic stress properties, salinity tolerance, drought tolerance, temperature tolerance, pesticide tolerance, etc. And then we made certain consortia. We just studied single microbes and then consortia on the production of wheat, and we found that some of them are working very well and giving good growth and productivity of wheat. And then we made a consortia and its granules, organic granules, which are which consists the microbial consortia, and we applied in experimental field, and we have found that the formulation of these exotic microbes which we prepared by us have given the productivity of grain and straw more than chemical fertilizer, standard, rather the standard dosage of chemical fertilizers. Then we designed a kind of above ground combination of various plants, perennial and annual plants. We used Indian licorice, stevia, and chili above ground, and below ground we used different combination of these microbes, and we could see a lot of benefit in crop production. Net economic benefit has been very, very significant, and say total dry biomass in the year has increased, plant bio-dry mass and organic soil organic biomass also. All soil properties has been improved very significantly. Then above ground and below ground carbon sequestration has been estimated, and it has been found very, very significantly improved due to increasing species diversity below ground and above ground both. Well, these certain basically when we go for certain when we go for any cropping system, we have to see that biomass is one thing, but then there is one part which is important for its yield and economic gains. So that part especially has been required to be assessed, and here this licorice has contained glycyrrhizine acid, which is important, and this has a good market value. So we studied it, and we found that by adding microbes, we could increase the glycyrrhizic acid content in licorice. Then some perennial aromatic grasses of high value cultivated individually or marginal land, where, as I told you very in beginning, that the marginal land has all these grasses, otherwise also. We studied their various combinations and tried to understand which kind of species diversity will be required above ground and which kind of microbial input can be helpful. Here, for this experiment, we used local native microbes from the soil of these were isolated from soil of vetiver, and then their combination were prepared, and then we used those microbes for nutritional and other property, and we could see that these aromatic grasses in various combinations and alone can be grown well with microbial inocula on very low input and only organic input of microbial biofertilizers. If you look on the growth and productivity, the final harvest, second harvest, and third harvest, first harvest, we made 3 harvests in a year. And here you can just see that all parameters were highly improved due to addition of these microbial parameters. Here, plant height, number of slips, leaf area index, fresh biomass, and dry biomass, all those parameters got very, very significantly improved. Then we made different kind of combination of these plants to develop, to assess the impact of species diversity in platted experimental plants. And we could see that it is not always that if you increase the, here you can see, let us discuss data given in last column, that is 12 month and with PGPR. It is certainly vetiver as single culture has maximum soil microbial biomass carbon because it has a very good rhizosporic nick. But and there are some decrease in certain formulations, but even you can just see in formulation of vetiver and lemongrass and pomarosa, there had been higher level of soil microbial carbon biomass. It is not just related to increase in species diversity of above ground cropping system, because where the in last column you can just see where all 4 grasses were used together, then the total carbon soil microbial biomass carbon gone down. But certainly it is more than whatever there without plant growth promoters. So plant growth promoters certainly helped in increasing its soil microbial biomass carbon, its carbon sequestration, and also in its total productivity. So here we have shown the soil carbon sequestration in all 4 vetiver, lemongrass, pomarosa, and citronella, and we have seen that the carbon sequestration in terms of plant biomass is certainly it is again vetiver is it all depends on the type of plants, means canopy of plants, and vetiver has a big canopy than others. So the plants having vetiver in its combination are showing better aerial biomass, but certainly the species richness has increased the plant biomass also in various systems. So soil carbon sequestration is more. Aerial is depending on the plant type. The PGPR consortium and species richness were also assessed for yield, oil yield, and economics of perennial aromatic grasses, and we have seen that oil content was also improved by plant growth promoters, microbial plant growth promoters, and in certain cases there has been significant increase. Oil yield has increased. You can just see without these microbes, it was about 13.19 and 16.38. The value is increased if these microbial promoters were added. So it impact oil yield and cultivation cost, basically cost of grass return. Grass return was high and net return was high. So I just here I want to say that it is possible to design below ground and above ground systems, agroecosystems, and through that we can increase species diversity and we can increase the economic benefit also just by non-chemical means. Major component of essential oil were not changed much because that is also important for certain crops that their nutritive value or their say commercial value should not get changed. If that will change, then value of the product will get get reduced, and whatever we are planning may not work that way. Okay, then this was published in a paper, Land Degradation and Development, this part. Then we published some 2 more papers on these aspects. And then in conclusion, I can say that ecological restoration requires reimagination, recreation, and re-establishment of novel ecosystems for sustainable agriculture, afforestation, land and water decontamination, reducing man-made conflict of producing food crop, commercial crops and energy crops, etc. from the fertile agricultural land, and tribals, the conflict of tribals with governance system. We can regenerate economically and ecologically useful below ground and above ground ecosystems in the wasteland on low expenditure, which will convert carbon sources into carbon sinks, which will give us enormous economic and ecological benefits. So these are some of my co-workers related to this work: Dr. Kripal Singh, Dr. Asutosh Avasthi, Pawan Kumar Madhesia, Roli Mishra, Pradeep Kumar, and Pawan Kumar. And with this, I am just closing my presentation, and I shall be available for any discussion. So thank you for your attention.
Yeah, thank you so much, sir. Yeah, now it will be the open for the question and the any query. The participants can ask.
Good evening, sir. May I ask a question?
Yes,
sure,
sir.
please.
Sir, you have talked about crop production in the starting. So we know that in Indian Himalayan region, yield is decreasing, sir. And they are doing substantive farming practices. So, sir, despite all the favorable conditions there, why is it so, sir? Like, how can we improve the crop yield in that area, sir? In the Indian Himalayan region?
Basically, the degradation causes loss in fertility. The degradation in any agroecosystem by various anthropogenic regions causes loss in soil diversity, soil fertility, and that can be restored by using, say, regenerating the microbes in that area or adding microbes in that area. Then for basically now there is change in agroclimatic conditions. So whatever agroclimatic conditions are there, one has to, and need of people, need of market are also getting changed. So we have to design the above ground diversity, crop diversity, and below ground diversity has to be regenerated. And this I wanted to propose that one can go for designing new kind of agroecosystem there, below ground and above ground both. So this way we can restore uh, the the agroecosystem, and we can start again plantation there in degraded areas.
Okay, thank you, sir.
Yeah. Any other query from anyone? So I just okay. So I think that there is no question. If you any question in the future, you can ask and write to the sir in the email.
Yeah, welcome.
So now I again moving towards the formal vote of the thanks to the sir. So I request to the Professor C. Rajsekharan to give the vote of thanks to the sir. Yeah, and yes, and welcome to the Professor Sodhi from the Rajiv Gandhi National Institute of the Youth Development. Welcome, sir. Over to you, Rajsekharan, sir. And Dr. Y.B. Singh is also joined this session. Tomorrow he is our second speaker. So welcome, sir. Rajsekharan, sir, you are the firstly unmute yourself.
Yes, yes.
You are muted, sir.
Yes, yes, thank you. Once again, good afternoon to one and all. I am my immense pleasure to propose word of thanks to our distinguished speaker, Professor R.P. Singh. In this afternoon, he has given wonderful lecture related to what is the ecosystem, how to restore ecosystem, especially wastewater ecosystem, wet water ecosystem, then followed by the degraded wasteland, then designing and restoration of urban ecosystem, then bioremediation by using microbial means of growth and plants, that is a phyto remediation, all kind discussed. And the ecosystem economic services, it's one of the promising area for the next upcoming area. Then finally, he discussed about biofertilizers. That is how we can use our biofertilizers to enrich our ecosystem. It's a really amazing lecture for this afternoon, sir. And I'm behalf of our participants as well as organizing committee. It's my good pleasure to thanking you in this forum also. Behalf of our sponsors and behalf of organizers, I am highly thankful for your brainstorming lecture for our participants. Sir. Thank you very much, sir.
Thank you. Thank you very much. Thank you.
Thank you. So we are successfully conduction of the first day session. So tomorrow we will be rejoin for the session at 10 o'clock in the morning, and the two session is the every day, and every session is having the two lectures. So I just remind the first session is the tomorrow the 10 to 12:30, and second session from the 2 p.m. to 4:30 p.m. And meeting link will be the same for the entire 3-day sessions. So there will be no change in the meeting link. Yeah. Thank you, Sodhi sir, and thank you, YV Singh sir.
Thank you very much. Thank you, sir. So nice of you. Thank you very much.
Yeah, sir. So tomorrow we will be good session.
Yeah.
And everyone is excited to see how we can convert the municipal solid waste into the organic waste. And
I'll try.
also the working in the large area, the organic carbon and organic farming. So that lecture is wonderful and amazing. And our convener, Dr. Kalyani Krishna, is also want to talk to you. I think
Okay. I'll try my level best.
Yeah, sure, definitely.
Yeah. Okay. Thank you very much. Yes. Thank you.
Professor Sodhi, how are you, sir?
I'm okay, sir. I'm okay.
Yeah. After a long time,
long time.
That's really,
we met in Bangalore.
Yes, sir. Long ago, sir.
3
years. Bangalore, 2018 September. We had an interaction.
Yes.
And, only over mails and phone calls because of pandemic. Even though you are nearer to me or myself or yourself, we are not